Hey, welcome. Come on in. Hey, guess who got a new toy? That's right. To try to make these things a little bit better, to try to make these things a little more personable. The whiteboard with the microphone was good enough, but we went the extra mile, and yeah, saved up my allowance, saved up my lunch money, and I got myself a little webcam. So it's been a few weeks, almost a month since I've gotten any Pokemon cards, but eventually we'll get some more saved up so we can get back into that. But hey, welcome, and it is great. I should say, I don't want to say see you, but it's great to connect you with like this. Hey, you can see all the Chiefs gear, the Royals gear back there in the background there. If that's distracting, sorry about that. But that's the way it is. So hey, well, wow, what a great chapter to unveil the new toy in, and the plasma membrane. Man, this is an amazing chapter. Love talking about this stuff. But yeah, here we go. So now, instead of saying welcome, hey, Nevada High School students, bio students, I see you right there. Not dual credit, but Bio 2 Crowder students. Hey, I see you as well. So thank you for opening this up. Thank you for doing this. And man, we are in for a great talk. Great chapter, chapter 5, plasma membrane. Man, love talking this. This is probably, I don't know if it's my favorite organelle, but man, it's a super important one. And hey, now you get to see the air quotes that I always tell you about. So that'll be fun for us. But hey, let's get into it. And, the forehead's all washed out there. I know that. Maybe I should get into some of that makeup stuff like the TV people do. I have to talk to my wife about that today, but I'm sure I can't borrow none of hers. But I have to get my own, I guess. It wasn't that big exciting. A little 30 minutes. Of course, I don't even know. Maybe it takes longer than 30 minutes. So I got to plan more. But yeah, the forehead's pretty washed out there. Because I also got me a new lamp. Because the other lamp or light system in here just wouldn't have been enough. It's too dark, too grainy still. I was looking like some old Bigfoot footage. And some of you don't even know what Bigfoot footage is, so you have to Google that and you'll see what I'm talking about. But hey, welcome. Man, this is going to be exciting stuff here. And so with that, hey, let's get started. Chapter 5, we're going to break this up into 2 videos. This is pretty funny. I like being able to do that and see that. That's pretty cool right there. My wife had a different impersonation. Her thought was, those kids don't want to see you. And you're probably right about it. She's probably right about that. But it is what it is. So here we go. We're going to break it into 2 videos. The first video, the one you're watching now, will cover the basic structure and setup of the plasma membrane. And in part 2, we'll cover, in part 1, we'll cover the structure and passive transport. And then number 2, we'll cover active transport, and then all the I don't want to say miscellaneous, but peripheral information over this material. So with that, hey, bio students, Nevada High School, Crowder College students, let's get started. First thing, obviously, I need to scoot this over just a bit there. There we go. All right, so the first thing I want to do on my list, you know what I mean by list, I got myself a little note sheet here, make sure I say everything, make sure I talk about everything. So first thing I'm going to do is introduce all of the players. So let me get a little ink tray here, and let's see, how about, I don't know, what are we thinking? Well, we got so much reference to water over there, so let's just go with the blue. Okay, can't go wrong with blue. So we're just going to go around the table here, around all this, and we're going to talk about each of these. So we're going to start here. We'll just start here in the lower left-hand corner. Let's go a little bit thinner than that. Let's go all the way down to here. There we go. So peripheral membrane proteins, recognition. And this will be my next thing I got to focus on is getting a better writing situation, recognition. But these peripheral membrane proteins, and when we say that, we're talking about obviously these are the ones here. These are just on the edges. You can see them here. Here's another one, and then these little cats back in here. That's what they are as well. All these little guys, and probably not that one. That's a gated one. So, but they are about recognition. One of the things that we mentioned in Organelles, chapter 4, is that part of what your cells do is to recognize self. And when we say that, we're talking about, we mentioned that when our cells make something, there's a little marker put in it that says, hey, that's us. It's like all of our stuff has a certain symbol, okay, that means, hey, that's us. And so part of these peripheral membrane proteins, and we're going to look at other pictures of them, but they recognize self. So in other words, if a bacteria cell shows up, a virus shows up, which is obviously in the times that we're in a super important topic, but these peripheral membrane proteins are able to recognize, hey, that's not us. We didn't make that, and that cannot come inside. And you can knock on the door, but we're going to go, no, cannot come inside. So that's membrane or peripheral membrane proteins. So now let's talk about integral membrane protein. And again, we're just kind of introducing them, but we'll talk more about them individually at a later time during this broadcast. Oh, I'm having a great time with that, seeing that picture. I am enjoying that. Hopefully you are too. Of course, you probably have already taken a Post-it note and put the Post-it note all over this picture right here. And if so, I can certainly understand that. A little 3x5 Post-it note covering up the picture. I get that. But integral membrane protein, these are passageways. And I'm just going to put the word passage, but you'll recognize that I said passageways. You know what? I'm going to go all the way, and I'm going to do it all. Passageways. These are the things that allow things to come in and out. So if it's got to come in, or it's got to go out, either way, those are the passageways. You can think of them if you've ever been somewhere, and I haven't. We have overpasses, but that's not really the same thing. But if you've ever been somewhere where you have to, the highway that you're on, the road you're on goes through a mountain, and you got this tunnel to go through, it's something like that. Or if you can think of a pipe, the water comes from outside of your house into your house through these pipes. That would be kind of what these proteins are. They are passageways for things to come in and out. And there's a lot of ways that happens. And when we talk about diffusion, when we talk about facilitated diffusion, when we talk about active transport, you'll see some more of that. But lots of ways it happens. All right. The next thing we want to talk about, cytoskeleton filaments. We mentioned these in organelles. But these are structure. These are support. I'm going to put the word support here. But these are support. Okay, these simply is help the cell, the membrane, to kind of have its structure and all that kind of good stuff. Our next member is, our next player to talk about is cholesterol. Now, we often in the news, health news, whatever, you hear about cholesterol, bad cholesterol, good cholesterol, too much cholesterol. But your body does need, certain levels of cholesterol. And in this case, it is, what's one word I could put? I'm going to put structure. But what's really going on is the cholesterol is helping the tails. Let me finish this. It's like I can only do one thing at a time. These fatty acid tails of the phospholipid, and we're talking about these things right here, help to keep them where they're supposed to be. Help to keep them in place so they don't get tangled up, so they don't get out of whack and get in the way of the diffusion and things like that. Protein channels. Okay, these are super important. Again, these are passageways. Okay, but they have a bigger, so I'm going to put a little arrow over here too. They're a little more complicated because they have to recognize the molecule. They've got to recognize what's going on. They've got to be able to see what's going on, recognize the molecule or the ion, whichever it may be, and then they allow it to come in. It isn't just they're not a tunnel where anything can go through. They're not an open passageway. They have to recognize what's going on for it to happen. That's some good glare on the glasses right there, so you can't see my eyes. And maybe that's for the better. Maybe that's helping you get through. But it's the idea of recognition and being able to decide yes or no. It can come in. The next part, the phospholipid bilayer. I'm not going to put anything here. I'm just going to circle it and say that's this 2-layer system. You know what? I guess I am going to write something down here. That's this 2-layer system. Okay, 2 layers. You've got 1 layer here, and then you've got 1 layer here. The exact opposite, and we'll look at a close-up of this and talk about this. But it's got the heads on the outside, inside, away, and then the tails in the middle. And of course, there's all kinds of bonding going on and things like that. The next thing we're going to talk about is the glycolipids. Okay, the glycolipids. Make sure I'm getting these two, glycoproteins, glycolipids. The glycolipids, immunity. I don't know why I even paused there. They help with immunity. Both the glycos are about recognition. The glycolipids help with the immunity. So when they recognize something is not us, they can alert the immune system, the T cells, the macrophages, that kind of stuff, and say, come on boys, get over here and take care of this thing right there. And then the glycoproteins over here, they help with just recognition. What's one thing I want to put here? They help with recognition to decide if it can come in or not. So again, I'm just going to put recognition up here as well. And they are about should that thing be allowed to come in, that molecule or whatever has presented itself to us. So that's the main players. And again, most of the stuff that we will talk about individually as we go through the rest of this presentation, and all that stuff is down there somewhere. So we will get to all of that. All right, so hey, that's the players, that's the individual components of that. And because the membrane isn't one solid thing, it's a beautiful constructed organelle that has so many components, and they each have an important role. They each have a great role. They each have, in their own way, in their own special way, contribute to what's going on. But let's talk about some things first. Let's talk about the plasma membrane in kind of general terms. The words that are associated with, and I made that kind of big, so I'm possibly going to run out of room of everything that I want to say. The way that, and that's supposed to be an O, so let me finish that up there. There we go. I think Thought for a second that would have been the sea there. And that's supposed to be fluid mosaic. And that's what these pictures are representing here. When we talk about the plasma membrane, we emphasize the word fluid, not just because it's watery, okay, but fluid as in not solid, moves around, kind of like a motor oil is fluid, but it's got some thickness to it. It's got some structure to it, but it is fluid. And so that's what these pictures of the ocean. And when I was trying to pick out a picture, I couldn't decide between the two. So you know what I did? I said, you know what? Let's just put them both in there. Can't decide, so use them both. Pretty pictures, beautiful pictures of the ocean. Nice wave. I've never been. I mean, I've been to the ocean multiple times. Oceans, I should say. But never in a situation where I've seen a wave like that. That'd be pretty awesome, too. I mean, that's the kind you see in movies when the surfers are involved, or maybe a storm's coming in or something like that. But and then, of course, this is inside. You're down in the ocean looking up at the sunlight, and you see the different levels of light right there. But also mosaic. You know what a mosaic artwork is? This is an example of mosaic artwork. This is a picture, obviously, of a dolphin. You got the ocean down here, and then the air or whatever. It's jumped up out of the water. But what this mosaic artwork is just a collection of maybe broken pieces of glass or broken pieces of pottery that have been by color put together. Oh, that's one of the things I got to watch right there. I just scratched the head. I can't be doing that with the camera here. So I got to watch that kind of stuff. The fluid mosaic, it's just a collection of broken glass or pottery or ceramics or whatever, but they're just arranged by color in a way that they make a pattern. And the plasma membrane the same way. It's a combination or a organization of different parts designed in such a way that it works as an organelle. So that is the fluid mosaic, and the word model should be back here as well. Okay, so something else we want to recognize as I look over at my list here. It's asymmetrical. And I'm really going to go on a limb here because I didn't write that word down. I wrote down it was not identical, and that's not a very fancy word. So hopefully I do this right. Asymmet. I'm going to sound it out. Asymmetrical. Ooh, I think I'm going to do it. Oh, that's a bad looking C right there. Asymmetrical. In other words, it's not the same on the inside, the inner half, the membrane, as it is on the outer half. The outer half has a lot more security involved, a lot more recognition proteins, a lot more of these glyco stuff. This glyco stuff isn't on the inside. It doesn't need to be on the inside. It's supposed to recognize things that they're approaching. So a lot of recognition stuff on the exterior surface, and a lot more support and framework on the inside. So it's asymmetrical. It's not the same. If I split it in half, if I drew me a line all the way down through here, split it in half, the top half would not be the same as the bottom half. Okay, the next thing that we want to say is semi, and we're not talking about the truckers right here, although they are super important people, but semi-permeable. And that's a great science word when it comes to the plasma membrane, because it is, what it refers to is that things can come through. Things can come in, they can go out, and some can just do it on their own, diffusion, real small things, like water molecules, oxygen, things like that. But others have to have these passageways that I'm highlighting here. But so it is permeable. Things can go through, but the word semi refers to sometimes we got to okay it. Sometimes we got to sign off on it. You can think about think about a military base. And in all the movies that we've seen, you got the guy at the front. There's a gate, and the gate is closed, but there's a little shack out there. Guy standing inside of there. You got a little clipboard. You've got cameras, monitors. You've got phones, walkie-talkies. I'm sure heavily armed as well. And you pull up there, and he goes, "What's up?" And you go, "I'm here to see so and so, or I'm here to do such and such." And gets the old clipboard, checks on it, see if your name's on there. If your name's on there, or he just checks your ID badge to make sure that you have the security clearance to enter. And if everything checks out, he pushes a little button and the gate opens up and you go on in. But he also, if he checks out your security clearance and you are on a list or you don't have the clearance, he can also say, no, you got to turn that vehicle around. You got to go somewhere else. You don't have to go home, but you can't come in here. So that's semi-permeable. Okay, the plasma membrane can decide if you're coming in or if the molecule is coming in. So that's semi-permeable. Let's see, what else? It controls going in and out. I said that many times. I talk about immunity, talk about recognition. So let's talk about some special ways in which the membrane. So I'm going to close this and I'm going to move this up a little bit. We're going to do something a little fun here, and then we're going to start talking about these pictures. But let me get this. I did this line here just to kind of see what it looked like, and now it's in the way. So I'm going to get rid of that right there and then go back to my blue. No, you know what? We're going green because what I'm going to draw you needs to be green. And when I draw it, you will see, because we want to recognize that all cells have a membrane. Okay? All cells have a plasma membrane. All cells have the ability to control what comes in and what goes out. Okay, even bacteria cells have a membrane. That's how they can get their stuff out so quick and into our cells. But a special thing, okay, even if a cell has a cell wall, okay, it still has a membrane. So let's take an example of that. And you may say to yourself, man, how in the world would that work? So what kind of cell is that? Okay, what kind of cell is that right there? Hopefully, hands are going up all over the place, and everybody is screaming out, plant cell. Okay, and you may think, okay, so a rare situation. No, there are more cells with a cell wall than without a cell wall. So even though, okay, we're animals, we're super cool, we think we're top of the hill, when it comes to numbers, no, not so fast, my friend. We are a distant in the category of cells that have a cell wall compared to cells that do not. A lot more cell wallers out there. And let me get a different color here to do on the inside. Oh, let's go with this yellow right here. So you got this cell wall, and so just inside of that, okay, you're going to have the membrane. And when we look at photosynthesis, this will come into a big play, because obviously plant cells have a cell wall, and then they also have a membrane. So let me draw a little arrow out here. So this is the PM, plasma membrane. And then go back to my green. And then this is the cell wall. Now you may ask yourself, and it would be an obvious question if you did, if the membrane is what determines if things can come into the cell or not, how in the world does it get through the cell wall, okay, to the membrane? And that obviously, the cell wall obviously is permeable. Okay, it doesn't really make any choices. That's left up to the plasma membrane. So things can come through the cell wall all at once. The cell wall is support and structure. and then once it gets to the membrane, then that's when a decision is made whether you can enter or not. So there could be some things that hang out in between these two that we don't necessarily want in there, and then we just rely on the immune system or all that stuff to come along, the T cells, the macrophages, all that stuff to clean up that mess. All right, getting a little thirsty, time to take a drink, so here we go. First, glass of water on the camera. So there you go. How cool was that right there? All right, so that is all of that. So what we're going to do now is close this out. Let me go back to blue here. And we want blue, but we want that thin. So now we can close that out. And now we're going to go, I got to close this because I want to come back over to here. And so now we're going to kind of get into the individual components. Excuse me. Individual components of, see how I did that? Yeah, get out of the camera when you're going to cough. So I learned that in broadcasting school. So now we're going to talk about the individual components with everything. Now we've seen this picture before. We saw this picture when we did lipids way back in chapter 3. I mean, way back in chapter 3. This is chapter 5. So that's for Crowder people. That's a couple weeks ago. For high schoolers, a while ago. So you can see here it's September 6th. Yeah, tomorrow's Labor Day when I'm making this. But it's the idea of the phospholipid. And so just kind of break down and remind ourselves about this. This is obviously the head here. Okay, and it's polar. Meaning it associates with water. And when I say associates, it orientates itself towards the watery surface. And even so, that if you could go onto Amazon and buy yourself a bag full of phospholipids, and I don't even know if that's I always use the example because it's in the textbook and stuff, and it's actually a question in the back of the book. But if you can go on Amazon and buy yourself a bag of phospholipids, maybe they come in a big box and whatever, and you dump them into a glass of water, they would orientate themselves, okay, in such a way that the heads would move towards the watery environment and almost in that circular standpoint, and the tails would be trying to point away from the opposite direction. All right, so that's the head, that's the polar. Of course, down here, we've got the tails, I should say, and they are nonpolar, meaning they don't associate with water. And you got the whole hydrophobic, let me do this first, nonpolar. Okay, you got the whole hydrophobic, and that's right there. Hydrophilic, right there. Water loving, water fearing. Good words to give you an idea of what's happening, but certainly not water fearing, water loving. Just means it associates, doesn't associate. And then the key to all this is this middle part right here. Glycerol. And when I said the word glycerol, hopefully you had a little flashback again to chapter 3 and the lipids, because we've seen that word before. We talked about that in a couple of ways. And this is, I don't know if it's the exact picture, but it's almost identical to the one in the textbook right here. If you turn to that page, there would be this picture, and then right above it would be another picture showing you the glycerol and the fatty acid tails by themselves. And we'll certainly look at that in class. But glycerol is kind of the anchor for this whole thing. The phosphate group, which makes the phospho part of the phospholipid, attaches to it, and then the tails attach to it. And of course, we've seen this picture. So if we were to break this down, this CH2, okay, is right here, CH2, and then this CH, okay, is right there, and then this CH2 is down here. So you got your glycerol backbone, and these are the fatty acid tails, and that's what these are. And in this case, we just need two. This is a triglyceride. So officially, this would be a diglyceride, but it's a phospholipid, and if you incorporate the whole thing. And you got this little kink in here for the unsaturated, and that's super important. That keeps the tails from getting tangled up. Cholesterol also helps with that, but this also is a part of that structure as well. So that's the phospholipid, and let me close this and go back up here just slightly. So one of these, and then I got to bring my pen back up here. One of these is what one of these little things here are. So you can see that a phospholipid bilayer would have, a bunch of them, thousands and thousands of them, all lined up like little soldiers doing their job to keep us, to help us to receive what we need, and also does its part in holding on to the proteins to keep the things out that we don't need. And so, before we go away, let's talk about what's going on here, and what's going on here, and what's going on here. I've got an element being held together, or being connected to this fatty acid with this oxygen right here. And hopefully, just by me saying that, flash bulbs all over the room are going off, hands are going up, people are screaming out, dehydration synthesis has just occurred there. You've got these two parts. I got this part and I got this part that are connected, and so at one point in time, the glycerol would have had an OH, the fatty acid tail would have had an OH, and the H2O left the building, and what remained was the oxygen, the binding element. So I would have had the OH over here, and over here, I didn't finish my line there, and then of course over here we'd have had the HO, because the oxygen's always up against the bond there. And then these two and this would have left, and that would leave the oxygen to bond with the two carbons. Dehydration synthesis. Talked about it a bunch already, and there's another example of how it works. All right, so let's close this, and as I mentioned, all this other stuff is down here. So let's slide this up. Usually I take a look at the clock, and sometimes I even write it down if I'm really paying attention. I'm going to go up a little bit more here. And so I can kind of keep an idea on time, but I forgot to do that. I guess I was too excited about the camera. And besides, now that I got a camera, I know you want to see as much of me as you can, so we won't worry about the time, and we'll just enjoy this together. So now what we're going to do is go through and kind of talk about individual parts. Okay, the first thing we're going to talk about is, I'm going to scoot this over just a little bit so I can write over here. We're going to talk about these proteins. These are the proteins that we saw. I'm getting my mouse off my mouse pad here. The proteins that we saw within the phospholipid, or within the bilayer. And I got to remember E before I in this one. The proteins. So there's the two types. These are integral, meaning within. And these are peripheral. And these are probably words I should have typed, but They're just one word, so periph. Oh, I'm leaving something out there. I got that word written down. Oh, there should be a P right there. There we go. Peripheral. And that may still not be right. We better go take a look. Let's go back up here and take a look. It's up here. Isn't it? Yeah. P-E-R-I-P-O. Man, I left a bunch out. Alright. P-E-R. I better write it. No, I got it. P-E-R-I-P-E-R-A-L. And I butchered that. P-E-R-P. So let's just try this again. And you go over there. In fact, let's just kick you out. Alright, now we're ready. P-E-R-I-P-H-E-R-A-L. So, today's spelling lesson was brought to you by the good friends of... I was going to try to do a Sesame Street thing there, and I lost it. Oh, now that I think about it, there's supposed to be a G in there, integral. Well, I'm killing this one right here. So this is going to have to be our little secret that I butchered both of those words. That'll just be between you and me right there. All right, so integral. Okay, so these are, and these yellow boxes that you see here and that you see here, those are to represent the plasma membrane itself, and it gives you a frame of reference to what's going on here. And most of this we've already written down, so I don't think we're going to do any really writing. We've got it, it's up there, and but feel free to write it down as I say it. That would be a good idea by you. But integral proteins, these obviously span the entire length of you can see here, of the membrane, and they are passageways. Okay, these are things that whether they have to recognize it first or they're just gated open channels. There's a lot of variety there, but they are integral, meaning they are passageways. They let things in, they let things out, whatever's going on there. And that is their part. Peripheral, so I probably should just put the word passageways here. I'm just going to put passage of it. I mentioned it earlier, so I'm just going to put the word passage, and then I'm going to put the word, or the letters, R-E-C. This is recognition, and again, we already wrote this down. So recognition proteins, and again, they are on the top edge, so you don't see any on there on the bottom, because these are about security. These are about recognition. And so whether they have to recognize particular molecules, particular ions, they just have to, so it's got to be recognized. If it's not recognizable, or if these proteins decide that, hey, that's not us, so it's a no-go, abort mission, all that good stuff, that's their part of it. Recognizing, is that something that we need to be brought in, and then allowing it to happen. All right, so that is the proteins, and now we're going to get into, and I'm going to do some adjustments here. So let's go in here towards the bottom, and we're going to just start moving stuff down here. So you are going to get a little preview of what's coming up, and I don't know if you know enough about your biology to recognize what's the top of that one, what all this stuff is, but if you do, then you'll know what's coming up. And that one needs to be a little bit bigger, I can see. And bringing this one down. Let's go ahead and make it a little bit bigger. And bring this one down. And bring this cup of tea down here. And we're going to slide all this over. And now we will start typing, because this stuff, we're going to do a bunch here. And we just need to make sure that you are, you can read it. Because what we're going to talk about now is And yes, I gotta look down at the keyboard. I am not a typist by any means. And here goes glass of water or drink of water number 2. Not a typist, but no, I'll tell you what, I will try it. I will do that. Passive transport is our topic now. And the things, as I look over here, there are some key ways that it happens. So things we want to, and let's see if I can do this. Oh, no, I looked. Diffusion. Okay, we're going to talk about diffusion, facilitated diffusion. It's quicker if I look down there. And osmosis, which is a cool topic. It's kind of a mental challenge to understand osmosis, but we're going to work hard on it, and both here and in class, to make sure that we recognize what's going on. Some other key things to know about passive transport. No energy required. So in other words, for materials to move in and out of the cell with diffusion, it does not cost any energy. It just happens naturally. So what that typically means is that either the materials are small enough to do so on their own, or there's a passageway designed and built just for them. It's got to be recognized if that's the case, but no energy is required. And my eyes itchy, so I am going to bend down over here while I do this. Hopefully I got out of the camera. I didn't double check that, but no energy required. Down the concentration gradient. And that's a lot of science words right there. And what that just refers to is that for passive transport, things move from an area where there's a whole bunch of it to areas where there's very little of it. Okay? An example of that is this glass of tea over here. And let me get a little pen here. So we're talking this image right here and this tea bag here. Now, when this tea bag was put into this water, which I assume is hot water, initially the water was clear and the tea bag had all of the essence of tea within it. In other words, it was all in one particular area inside the tea bag. But over time, the tea diffused out or moved out from where it was, all of it, to areas that it was not. That's going down the concentration gradient from an area to where there's a whole bunch to an area to where there's none or little. It spreads out. Okay, gas molecules can do this as well, and this is how you are able to smell food cooking from another room, or how you can smell a skunk from up to a mile away that has sprayed. Because those air molecules, or those smell molecules, in their beginning were all in one spot, either from the oven or from the skunk, and over time they diffused out. And you can see they diffused out. That was some good hand gesture right there. They diffused out to areas where they're not. That's going down the concentration gradient. The opposite, when we talk about active transport, is where we have more and we take less and go up the concentration gradient. In other words, where we already have a bunch, we're going to add some more. And that example, but we'll do it again when we get there, is like a restaurant. Okay, restaurants may have a lot of food for today, but they've also got more food coming because they got a plan for tomorrow as well. They can't wait till they run out and then go and get it. It's got to be there because there's got to be maybe some prep work. You know, if you're a restaurant and you're smoking your meats and foods, that stuff has to cook for 12, 15, 16 hours. plus then rest for a day before it's ready. And so you got to be serving today, but you got to be cooking for tomorrow, that kind of stuff. So, but that's the next video. But it's the idea of understanding what down the concentration gradient means. It means simply that we have a lot, and it goes to where it's not. Okay, it's kind of like think about a rock. Let me draw this too. Then draw me a little triangle here, a little wedge. Okay, and I got a big rock right here. Okay, gravity is going to pull that rock down from where it is to where it's not, and you know it's going to go. This is the concentration gradient. More, less. It's going to go to areas where there's less of it. So what that means is for your cell, if you've got a bunch of, we'll say, sodium on the outside of your cell and very little on the inside, then sodium's going to come into the cell to try to balance out. Okay, that's the best way of saying it, trying to balance out. And of course it does so in these three ways right here. All right, so that is the background information for passive transport. So let's start talking about each of these three individually. And we'll slide this up a little bit here. And we're going to start with diffusion. Now let's go pen. Because we've kind of said everything, and especially these two bad boys right here. So this is diffusion. And see, now I can write and look up because I have to look at the screen. But this is diffusion right here. Okay, you can see that there is a whole bunch of whatever this molecule is here on the outside of the cell and very little on the inside of the cell. So over time, and you can see that right there, over time things are just going to drift in to the cell, and you can see them coming through here. Some already have, and this is like a story in three parts. This is the first scene, this is the second scene, and this is the third scene. So all out here, none in here. They begin to drift apart, and then once it's balanced, okay, or we've established equilibrium, and I hope you're proud of me, I spelled that one right. But that's in, even though that looks like a more challenging word, I've written that a lot of times, typed that a lot of times. So that one I know how to spell, that integral perfect. I don't know why I butchered those, but we did. All right, so we're looking to establish equilibrium with diffusion, and so these things happen naturally. They're small. They just kind of have free access to move in and out of the cell, because anything of danger or anything that we'd want to keep out would be a lot bigger than this, and would not be able to. So these things kind of have kind of have free range to come in and out of the cell at will. So that's diffusion. Okay, so let me close this, and so I will say we've only got the two. Oh, didn't mean to do that. Oh, because I don't have my pen. We've only got the two topics left: facilitated diffusion, osmosis. So we are getting close to the end, and I didn't mean to do that. Let's come over here and so we got 1, 2, 3, 5 pictures left to talk about, and then this one will be in the books. But again, you've got the ability, you've got the freedom to pause, stop, come back often as you need to, much as you need to. And let's do this one a little bit bigger here. All right, so now we're talking about facilitated diffusion. And let's get my blue up here. So facilitated T-A-T diffusion. And it follows the same rules of diffusion. The only difference is that's supposed to be an S. The only difference is it involves this protein. It involves this protein. Now it could be a protein that's just an open passageway. It's just always open, and things that can fit through it. So it differentiates by size. It's still going to be pretty small. Still going to be pretty trustworthy as far as size reference. Excuse me. But so size. So I'm back up. So I'm getting too many thoughts here. So it could be just an open passageway, but it could also be something that's designed for a particular ion, maybe sodium, Na plus, or chlorine, Cl minus. In other words, they've been involved in the bond. They've gained or lost electrons. That's the word ion. But so it recognizes those. When we get into active transport and we talk about the sodium-potassium pump, we'll see that. But some proteins are just designed for particular ions, elements, or some proteins are just designed for certain molecules. But in facilitated diffusion, still pretty small, still pretty kind of element-based, and it may be an open passageway, or it may have a little recognition marker on there to where, okay, your sodium, come on in. All right, your sodium, come on in. Oh, your sodium, come on in. Oh, your oxygen, no, your protein is over there, or chlorine, your protein is over there. That kind of stuff. That's facilitated diffusion. Still diffusion, but with help, facilitated. All right, let's close that, and oh, you know what? We're going to stay right here because I was getting ahead of myself. These aren't separate. These aren't separate pictures. Facilitated diffusion, I should have mentioned, let me get my pen here, by name. I told you what it did, but these are channel proteins. Again, passageways, could be open, could be recognition based, but those are channel proteins that do that. The opposite are carrier proteins. Carrier proteins are, again, facilitated diffusion, but they are designed to be a little more recognition based. They are designed for particular molecules. So if you are a carbohydrate and you want to come in, you've got a particular carrier protein that you go to. It'd be just like if you were a warehouse, or you're any company that's going to be getting a lot of truck shipments in, and say you handle water-based product, solid-based product. Maybe you handle food. So maybe you've got like seeds and things like that, and you've got beef, things like that, and you've got chicken, and you've got pigs. You're getting all these meats or feed for them, and you have the 4 doors. And so when a truck shows up, it's got to go to the right door. You can't have a load of chicken products taken to the seed or feed door. Okay, it can't work. You got to go to the chicken door. Or you're bringing a load of pork products. You got to go to the pork or the pig door and unload and get your stuff unloaded. Same thing here. If you're a particular molecule, you've got to go to the right, the proper protein is a better way of saying that. The proper protein. One, to be recognized, security clearance. Got a little check. Yep, you're on the list. Come on in. Or, nope, this isn't your door, this isn't your bag. You are not on my list, so no listy, no entry. Facilitated diffusion. All right, let me close this. And now we're on our final topic. So if you've stayed with me the entire time, wow, kudos to you right there. But if this is your second, third, fourth, fifth, sixth, seventh, whatever, part of the video or time to watch the video, nothing wrong with that as well. Last topic though, osmosis. Oh, forgot my O there. Osmosis. Now, the first thing to think about osmosis, if you just look at the definition, you see movement, and I'm just going to put the word move here, water. But that's a misnomer, because then students think, okay, osmosis is just moving water. No, that is not what that's not what the deep meaning of osmosis is. It is moving water, but to dilute. So diffusion moves all molecules except for water. Water moves through the process of osmosis. But it's not the movement of water for the sake of moving water. The water is moving to dilute something else. In other words, the water is saying, okay, hey, there's a bunch of sugar molecules over there, and there's not very many here. So most of us is going to go over there to dilute the sugar molecules. Some of us are going to stay here, but a lot of us are going to go over there to dilute the molecules. So let's look at our first example of that, and that's right here. This is a before, and this is an after picture. And you can see that here the water levels on both sides of this semi-permeable membrane, aka phospholipid bilayer, aka plasma membrane. So water can move through as it needs to, as it wants to. Now, if you'll also notice, we've got a whole bunch of whatever this molecule is over here and very little over here. So the water says, hey, we got to go help dilute that stuff over there. We got to go over there and help dilute it down. So osmosis is that this water is about to move, but not for the sake of getting the water from here to there, because we need it over there. It's to dilute down the materials. So some of the water on this side went to over here, and that's why we had these difference in water levels. Now, these little molecules, whatever they are, they're too big to pass through. So they need, so even though we put something in here that water can move through, this isn't a true bilayer, or else there'd be a way for this molecule to move. The molecule can't go back through. So the water goes over there to dilute it down. So hopefully that is clear what the true purpose of osmosis is. It does move water, or water is moved through the process of osmosis, but not to get water from here to over there, like, you know, there's a fire or something and we got to put the water out. But you know what? That would be the same thing, because it's going over there to dilute or to water down. So it's not the movement of water for the sake of moving water. It's moving water to try to dilute a product. Okay, and now our last two pictures. Here are some examples. And you know what? Let's go a little bit bigger here. These are, and let me clear this, and let's bring this up a little bit. These are scenarios in which you would find examples of. These are animal cells These are plant cells. And so let's look at these three words: hypertonic, hypotonic, and isotonic. Now, hypertonic is a majority of water is going to exit the cell. Okay, it's leaving. When I was growing up, and I joke, but there was some truth to it. When it was summertime and we were home, and my mom was like, "Oh boy," because me and my brother, I mean, we were. You wouldn't know it now when you look at me because I'm old and I just kind of stand around. I'm slow, but when I was, 8, 9, 10, man, we were all over the place, and we were outside kids, no doubt about that. And of course, I grew up in a different time. I grew up in the mean streets of Joplin, as you've heard me say many times. And in those days, 70s, we went outside and played, and there really wasn't a lot of concern about anything of danger. So even the point to where we decide we're going over to Billy's house, we jump on our bikes, take off, and so on and so forth. But when we'd go outside, we'd eat breakfast, and we'd go outside to play, because we were hyperactive. My mom would say, "Hey, you two boys, you are outside." And when we go outside, we swear to this day that every time we did that, we heard the screen door click, because she locked us because we weren't going to come back inside. No, she had no thank you. We were outside. So hypertonic, a hyperactive kid is sent outside. So the water, for the most part, goes out of the cell. Out of the cell. Now, what would cause this, you might ask? All right, we're thinking osmosis. If water moves to try to dilute something, that means that there's more, we'll say salt. Okay, there's more salt on the outside than there is salt on the inside. I'm putting a little bit of salt here because that's supposed to be represented that it's on the inside. So a bunch of salt on the outside, a little bit of salt on the inside, and so the water says, all right, we got to go help dilute that salt so it leaves the cell. Okay, that's bad. Okay, just look at these cells right here. That is bad. Spoiler alert. No, you don't want your cells looking like that. Now, what would be an example, a real life example of this? And I don't know if it's a thing anymore, but it was a thing when I was a kid, because we had gardens. My wife has gardens now, but she's got special, chemicals for it and stuff. And my mom liked to garden, and when she'd see slugs in her garden eating on her plants and mucking everything up, their slime trails, she'd go get the salt shaker. And now, I don't recommend you doing this because, slugs have feelings too, but I'm just telling you. So you'd pour salt on the slug, and it would essentially dissolve it, or it looked like it was just melting it. But what's really happening is that the slug cells are saying, "Oh wow, hey, there's a whole bunch of salt on the exterior of us. We got to go dilute that down." And when it left the cell to dilute it, the cells just shrunk up, and the slug died, reduced in space. So, excuse me. So that is an example of that. And we'll go over here to hypotonic. Okay, hypotonic. Now we've got a situation where the water's coming in. Okay, so it's the opposite of hypertonic. So now we got a little bit of salt out here and a bunch of salt in here. And I'm using salt because I used my slug example, but it could be anything. Sugars, any molecule. Okay, the water is going to move to try to dilute it down. So now water's rushing in to the cell. Now, and I should have put a out arrow over here. Rushing in, and for a while that's okay, because your cells do like a healthy amount of water. But like this bad boy right here, you get too much, and the cell's going to burst. And I know that sounds cool, but spoiler alert, that's bad as well. You don't want your cells bursting. What you do want is this scenario here. You want passive transport, you want active transport to properly be doing its job to where you got equilibrium or balance, to where the water can just flow nice and easy. A little bit goes in, a little bit goes out, that kind of stuff, and you got a nice isotonic situation right there, to where everything is balanced, everything is lovely, and we got no problems anywhere. So for our last picture, now we're going to look at a plant cell. This is obviously a plant cell because we got this rectangle shape here. Okay, and how it looks in these same scenarios. Now for us, two of them were bad. Either one of these two, okay, bad news for us. We want to be here. But for a plant, only one bad scenario, and that's hypertonic. No cell needs to lose all of its water. Now think about the structure here. This is a vacuole. Okay, we've got this vacuole here, and if the water all leaves, the cell shrinks. This is what happens when plants wilt because they ain't gotten any water in a while. They start to wilt over. The hypertonic situation. Again, the water's leaving to go dilute something. Okay, but a hypotonic situation for a plant isn't so bad because you can see what's happened here. The vacuole is filled up to be a nice, big, and puffy, and it's pushing against the cytoplasm, which is pushing against the cell wall, and makes this nice, solid cell right here. And that cell, when they're stacked on top of each other, acts as like a vertebrae for us, to keep us upright. See how I sit up like that? That's to emphasize that. That's pretty good. That's a pretty good teaching move right there. So to make that cell wall act just like a spine. Now, if it just continued to get water, okay, eventually maybe it does turn into a bad situation, but certainly handle a lot more because this cell wall gives it strength. But the perfect scenario, this part right here where you got this pressure up against you, that's called turgor pressure. Turger. And that's simply a word that references the fact that the vacuole fills up, it pushes against the cytoplasm, which pushes against the cell wall to create that structure. But of course, isotonic is the best situation to be in, where the water that's going out or going in is the same as going out, so everything's nice and equal and balanced. Wow, that was a bunch. And I don't even want to guess on the time. It felt like this is a long one. So if that's the case, much apologies, but a lot of stuff to talk about. And but again, hopefully you enjoyed seeing my beautiful smiling face up here. But again, thank you, Crowder College students, DeVita High School students. Appreciate you guys. Love you guys for doing this. Can't wait to see you again. Against Labor Day, so I got tomorrow off, and so we're going to enjoy that. Going to put some stuff on the grill, put some stuff in the smoker. We're doing that today. So Sunday, yeah, we're putting a nice ham in there. Let that smoke all up, and enjoy that tonight. And then tomorrow we got some stuff for the grill. Going to have a great time. Again, hope you are safe. I can't even think of how deep we would be into the school year when you're seeing this. For Crowder students, we're week 6 or 7, 8, possibly halfway. So we're looking early October-ish. The high school students, we're after Christmas because this is chapter 5. Yeah, so we're after Christmas doing this. Matter of fact, we're probably into February by now. So hopefully things are still intact and going well. But anyway, hey, enjoyed it. Looking forward to doing this more often. And with that, hey, we will say goodbye.